Hello everyone, in this video we're going to learn how to read the codon chart and do some practice. So as you can see, this picture right here is a codon chart that you've already received in class, so you can follow along using your own codon chart as well. When you're reading a codon chart, you're reading the mRNA codon. You're not reading the anticodon, you're not reading the DNA, it's the mRNA that we're reading, because on the ribosome it's the mRNA that gets read by the ribosome. The ribosome reads the mRNA 3 bases at a time, so one codon has 3 RNA bases, or specifically mRNA bases in it. So for example, if we want to look for AUG as an mRNA codon, and we want to figure out what amino acid is corresponding to that particular codon, we look on the left side first. That's your first base, right? So if we're looking for AUG, the first base is A. So we find A right here. And then the second step is you want to look across after you find the first base. So your second base is U. And as you can see, this first box contains everything A and U for the first and second base, right? Because the second, this is all U's, right? Because if you look at the second base right here, the second base U is right here. So AUG codes for the start codon. The start codon is a signal for translation to start, but it also codes for an amino acid itself, which is methionine. So if you are given a question that contains AUG codon in there, you want to write down the amino acid methionine instead of writing start, because really AUG codes for the amino acid methionine and is not only a start codon. All right, let's try another one. Let's try CCC, right? You start with the first codon, which is C. You look across, you find CC, and you find CCC codes for proline, which is another type of amino acid. So what you see here is that one codon, which is 3 bases on the mRNA, codes for one amino acid. But one amino acid can have multiple codons coding for it. Something else that you want to notice here is that we have something called the stop codon. So the stop codon is a signal for translation to stop. At this point, the stop codon doesn't code for an amino acid itself. So when you see a stop codon, you simply stop translating. Whatever is after the stop codon, you don't actually translate it. You just leave it behind. You stop right there when it tells you to stop. Okay? So there's a worksheet that you were assigned to do this week. We're going to take a look at some of the answers on the worksheet, and I'm going to walk over the process with you. So let's take a look right here. So the first few questions are pretty easy, things you've already learned. The central dogma is DNA to RNA to protein. Again, you want to avoid the urge of saying DNA is made into RNA, because that's not what happens, right? We have DNA code that codes for how we can construct RNA, but RNA and DNA are two completely different things. Same thing with RNA to protein. RNA to protein means we're using the RNA information to figure out how to construct this protein, this polypeptide chain, right? But that doesn't mean that we're turning RNA into protein. That's not how it works. RNA is RNA, protein is protein. It's simply, the RNA simply gives us the information on how to make the protein. So the next one, DNA is transcribed into mRNA in the cytoplasm. What did I just say? In the cytoplasm, if this were to be a prokaryotic cell, right? Because the prokaryotic cell doesn't have a nucleus. But you should say the nucleus if this were to be a eukaryotic cell. mRNA is transcribed into an amino acid sequence at the ribosome. So this is something that happens to everything. It doesn't matter what kind of organism you are. You have ribosomes that has a job to do translation. And ribosome is always in the cytoplasm. Okay, I don't know why the words are so small. You heard what I said. All that matters. Okay, so now, whenever you're given DNA bases, you should know that DNA is double-stranded, which means if you're given one DNA strand, you should be able to figure out what the complementary strand is. So let's take a look at how you can pair up the complementary base pairs for a DNA, right? If you're given A in one strand, the pair for it should be T. If you're given T, the pair should be A. If you're given C, should be G, and if you are given G, the pairing should be C, right? However, if you're pairing up RNA and you say, why are we getting RNA from DNA? That's what transcription is, right? You're given DNA code, use that as a template, and that's how you can figure out how to construct your RNA. So if you have DNA A, instead of pairing up with T, in RNA you pair it up with U because there is no T in RNA. And then if you're given A, you pair it with to T, you pair it with A. If you're given C, you pair it with G. If you're given G, you pair it with C. So the only difference between the left side and the right side, as you can see, is when you're given an A. If you're looking to build a DNA, you continue that with a T complementary to A. If you're looking for an RNA base, then you have to find the complementary base pair of A in RNA will be U. All right, so let's do some practice. So you have this DNA double strand. The other strand of DNA will be ATA, and then, ATA, and then this one will be, this is not working out. Okay. All right, this next one will be GAC, right? However, if you're creating an mRNA strand from the bottom DNA strand, right? There are two strands of DNA. Which one are we using? We're using the bottom one. So if you're using the bottom one, you have GAC, and that gives you CUG, right? Similarly, over here, it's asking you to figure out what the entire mRNA is using the bottom strand again. So that will be AUG, which is the start codon, which codes for methionine. Then you have GGC, AAA, UAC, GGU. Okay, so if you're given a sequence of some sort of nucleotide, you should be able to tell whether it's a DNA or RNA, because anytime you see T anywhere in the strand, that has to be DNA, and anytime you see U, that has to be RNA. Okay, and we're saying specifically this is mRNA because only mRNA gets translated into proteins. All the other RNAs are still built from DNA DNA codes, but only mRNA gets translated into proteins or amino acid sequences or polypeptide sequence. Okay. So now we have some practice problem. Let's do one together, and in our shared folder, I'll post answers to all the other ones. Except this is not working at all. Come on. All right, let's see. All right, here we go. Let's get rid of this one. All right, so here we have a DNA strand. Oh my God, what just happened? Okay. All right, so we're gonna start with the first one. T matches with A matches with U, C matches with G, and we continue on. We have G, C, U, G, 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 A, A, C, G, A, G, A, C, C, U, A, A, G, U. I really hope I'm right. Okay, let's take a look. A, U, G, right? T pairs with A, U, G, G, C, U, G, 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 A, C, C pairs with G, G, A, G. We have A, C, C, U, A, G, U. Okay, very good. Now, from this mRNA sequence, right, you always, whether the question tells you to do transcription first or not, if you're trying to figure out the amino acid sequence, you have to do transcription first, right? Otherwise, it just doesn't work. So now we're looking for the amino acid sequence. We have AUG, which is methionine, right? Which is right here, AUG. And then we have GCU, so we find G, we look across C, and we find GCU codes for alanine. And then we have GGGG, we look across G, we find GGG, which is glycine. And then we have AAC, you look to the left, A, look across, AC, so asparagine, ASN. And then we have GAG on the bottom, G, look across, A, G, glutamine, glutamic acid, GAG. And then we have ACC, left, right, ACC, THR. And then we have UAA on the left, again, U, look across, A. So here's the thing, you reach a stop codon, right? Even though we have UAA and then we have AUG behind it, because AUG shows up after the stop codon, even though AUG codes for another, AGU, which is right here, right? It codes for another amino acid serine, you've already gotten to the stop codon, which means you stop. And you don't have to write the word stop. Actually, don't write the word stop because stop is not an amino acid, right? When you reach stop, you simply stop. So by this point, this is all you have. That's where you stop, okay? Similarly, we have question number 2 and question number 3. You should be able to figure out how to write the mRNA sequence and how to write the amino acid sequence. If you still have questions after seeing the answer key, please find me on Monday morning. I hope this is helpful. Have a wonderful weekend.